some background on this topic.
Great. Thank you so much, Liz. I'm
Mhm.
going to share my screen. Okay, let's get it at the beginning. All right. Well, thank you guys so much for all being here, and thank you, Liz, for having me speak. As she said, my name is Catherine Farrell. I am the Aquatic Invasive Species Program Manager for the Finger Lakes Partnership for Regional Invasive Species Management. So we also call that the Finger Lakes PRISM. I am going to be starting our info session out tonight with just a little bit of background as well as history of hydrilla on Cayuga Lake. So I want to start off by describing my organization, just in case you've never heard of us. The Finger Lakes PRISM is one of 8 PRISMs across the state. And these PRISMs are funded by the New York Environmental Protection Fund and administered by the DEC. Each PRISM also has a host organization, and ours is the Finger Lakes Institute at Hobart and William Smith College. And this is a really wonderful host organization for us because we get to interact with research professionals who are dealing with watershed science, including contaminants, nutrients, watershed management, and then the PRISM brings in the invasive species programs. So our PRISM is quite large. We have 17 counties between Rochester, Syracuse, and Binghamton. So there are many water bodies for us to think about. I wanted to start off also with just briefly describing why we care about invasive species, what is an invasive species. They are any non-native organism that has entered a habitat and is causing negative impacts, either on the habitat, on the economy, or on human health. So the negative economic impacts can include problems with infrastructure and costs associated with that, or potentially the management of those invasive species can be extremely costly as well. And then invasive species also have environmental impacts, which we are considering the decreases in native species that are associated, as well as the surrounding habitat effects that could make the habitat less suitable for those native species as well. And then occasionally invasive species also have human health impacts. If we see changes to soil, water, or air quality, they can even have impacts on flooding or cause injury or disease or illness. And then we also we have a lot of connectivity within our region. So this is a good reason for us to really consider aquatic invasive species. So we have the Finger Lakes that connect up to Lake Ontario and are extremely naturally connected through rivers, streams, and creeks. And then we also have increased connectivity between these water bodies due to the level of boating culture as well as fishing culture within the region. So sorry, I'm getting notifications. I hope you guys are not seeing those. I apologize. So we have an increased level of tourism within this region, and this allows for the human movement of invasive species across the water bodies. And so we're seeing on the right here, we have a figure from our Watercraft Inspection Steward Program, where all the points that are on the outside here are boaters who were surveyed that said they were coming from Cayuga Lake. So what happens here is if there's an invasive species within Cayuga Lake, it can attach to any of these watercrafts and move on to these surrounding water bodies. And the invasive species that we are focusing on today is Hydrilla, aka watertime, or Hydrilla verticillata. So this invasive species arrived in the United States in about 1950 and came here due to the aquarium trade. So this has to do with individuals who have this plant in their fish tanks, and one or multiple people release them into water bodies. And this plant is native to Africa, Asia, and Australia. And in its invasive range, it has a broad habitat range. There are 2 types of hydrilla within the United States. We know them as monoecious and dioecious. So on this map here, you can see in the southern states, we have the dioecious form, and up in the northeast region where we are, we have the monoecious form. And the difference here is that the monoecious hydrilla has both female and male reproductive parts, so it can produce sexually as well as asexually. Moving on to the physical characteristics of hydrilla, we often get confused, well, not we, but some people often get confused with a native species called Elodea. But once you are aware of the differences, it becomes much easier to tell them apart. So hydrilla has leaf serrations along the edges of the leaves. So if you look very closely, you can see that they're kind of spiky along the edges. And then those leaves are in whorls of 3 to 8, usually about 5. And the stems of hydrilla, they grow very quickly and can get all the way up to 25 feet in some circumstances. So this very rapid growth can cause thick mats of vegetation in their established environments. And hydrilla can reproduce in multiple ways, which I've already mentioned. They can fragment, so if little pieces of hydrilla break off, they can float to new locations and establish roots. They also reproduce via seeds and tubers, and those tubers can remain within the sediment, and then they can pop up in the following season. And they also reproduce via turions, which are overwintering buds that pop off and can float to new locations as well. So as I've kind of mentioned, hydrilla is an incredibly impactful invasive species, and we see those environmental impacts here in this photo in the Connecticut River. It's a very inundated situation with hydrilla, where you can see that thick mat of vegetation that causes problems for native plant species as it blocks sunlight from entering the water body. We also see problems associated with boating and movement along the water body if those plants are going to get stuck in the boat motors. And in addition to those environmental impacts, we're also seeing an immensely expensive invasive species here, with these estimates in the top right for if hydrilla went unmanaged within the Great Lakes region. It could be up to hundreds of millions of dollars if nobody was doing anything about it. And so this is deemed the least wanted invasive species by the Council of Great Lakes Governors. So hydrilla has a pretty significant history on Cayuga Lake, and it started back in 2011 on the southern portion of the lake. We saw that Discover Cayuga Lake detected hydrilla on their floating classroom, and that is what started it all. And then in 2016, there was another detection up in Aurora. And we move on to 2018, where there was a detection in King Ferry. And in 2019, we found it down in Lansing. And last but not least, in 2022, it was found in Sheldrake. So we've included an overview here of the timeline of hydrilla detections as well as management of hydrilla. And you can see that it goes all the way back to the start. When it was detected, treatment was started immediately. And so in these early years, when hydrilla was located down in the southern end and down near Ithaca, there was a local task force that was managing the situation down in Tompkins County. And then as hydrilla moved northward, we had an increase in the number of stakeholders who were involved with this project. So as we saw hydrilla move over to Aurora, we had the presence of the United States Army Corps involved, as well as PRISM and DEC, kind of mitigating various aspects of this project. And some things to note here are that there are some years where we see no hydrilla detections, but then in the following year we see hydrilla again. So it's just showing it's essential to continue monitoring until you're certain the hydrilla is gone. And then something else that's notable is that King Ferry has seen relative success in getting rid of hydrilla in recent years, as well as Ithaca seeing some success in recent years as well. So as I mentioned, there have been a number of stakeholders involved in the Cayuga Lake hydrilla infestation. It started with a local task force in Tompkins County, and then it became a lake-wide task force, and even there's conversations in statewide task forces about the hydrilla on Cayuga Lake. And it's really awesome if you guys could stay involved. So we have lots of different ways that you guys can do that. So first and foremost, if you see hydrilla, please report it to the Finger Lakes PRISM. My contact information is on this slide. You can also report it to the DEC. And we ask that you take good pictures. If you think what you're seeing is hydrilla, we would like to be able to confirm it. And then also get involved with citizen science. There's lots of opportunities, but one example includes the Macrophyte Survey Program hosted by the Finger Lakes PRISM. And so I've also included here a QR code that you guys can scan and sign up for our listserv. We'll post all kinds of updates about our programs and our events. And additionally, you can attend other kinds of invasive species related events. We have a lot of water chestnut pulls throughout the season, and we would love to see you all there. So that is it for me. Thank you guys so much, and I hope you enjoy the rest of the presentations. Let's see.
Great. Thank you so much, Catherine. So if anyone has one question they'd like to jump in and ask, you can raise the little Zoom hand or unmute your microphone, and we'll sneak one in here as Rich Ruby gets set up. He's our next speaker from the Army Corps of Engineers and can work on bringing his slides in with the screen share. All right, Rich, I just sent you the sharing abilities. And just a reminder to folks as we're getting set up that these sessions are recorded and posted to Cayuga Lake Watershed Network's YouTube channel, so you can go back and look and listen to the talks. You can also view the slides, which we post online to our website, so no need to write everything down that you want to remember. It'll be posted soon. And it looks like we have Rich's slides available now, so I'll turn it over to you. Thanks so much.
Okay, great. Thank you very much. I want to thank everybody for attending tonight to listen about. I think this is good godless. It's probably our fifth, sixth, or longer annual where we've been actually providing a lot more public participation in this, which is definitely something that we enjoy, and especially hearing back from all the different stakeholders on the lake. So I'm going to just go over briefly some of the herbicides that we use out on the lake, and also then just talk about what we found or what we treated this year in Aurora and Ithaca, and what we found as a result of those treatments. Again, we did tweak them a little bit, as we usually do, given the fact that our program that we're doing this work under has been a research program that allows us to look at potentially treating hydrilla in these more difficult areas to treat, in these high mixing areas like the littoral zones of the lake itself, and then also in some of the canals in that. We do some work in the Erie Canal as well as the Niagara River in Western New York, where we have some hydrilla patches as well, or infestations. So I'd like to thank, or at least give a note to Mike Voorhees. He's sitting and hiding on the in the group here. He's taken over as the project manager. He's a biologist, and he's been on this project with me ever since we got going here in Aurora, which is about 2016, 2017. And then there also were was some outreach and was some technical assistance provided way back in the initial infestation, or discovery of the initial infestation down in Cayuga Inlet, by some of our members of our team from our Engineering Research Development Center and the University of Florida. As Catherine already provided, there's a whole host of different project partners on this. It isn't just the Corps, it isn't just DEC, there's PRISM, and there are a lot of local organizations. Cayuga Watershed Network as well has been great with helping get the word out about the issue we have with hydrilla, as well as helping us reach out and make sure that everybody's aware of the treatments that we're doing and why we're doing them. And then also we work very closely with not only the mayors and the clerks and the town supervisors, but we also work really closely with the county health departments, at least in the two areas where we do, both Tompkins County as well as Cayuga County, making sure we post our results up weekly with where our treatments are, and go ahead and also help with any outreach that we might have. So we'll talk about, like I said, the overview of the herbicides, the treatment and water monitoring that we did this year, as well as we do what they call point intercept surveying, which is where we actually have a known, or we have a grid of points, and we go to those points throughout the season, throw a rake and a sample to be able to identify the types of plants that are out there, as well as get a relative abundance. In addition to that, we actually do hydroacoustics with our sonar, and we collect that information, and that gives us more quantitative information on what's going on with the plant biovolume, not only within our treatment areas, but also outside of our treatment areas. So the goals have always been with this project is first and foremost public safety. Whatever control method that we were going to use, and there are a host of different ones that oftentimes are utilized in aquatic plant control. Fortunately, hydrilla is a very unique an interesting character, and it's one of the more aggressive and invasive plants that we currently are dealing with, and as a result, it really limits, because of some of the limitations we have with the environment where it's in, as well as the way it spreads, and that we oftentimes are limited to our herbicides. And I know Emily's going to talk a little bit more detail about all the different methods in her talk right after mine. So the second thing is one of the reasons we're worried about it, Catherine's already touched on that, is wherever this gets in, we're trying to potentially protect the amazing resources that we have within the Finger Lakes themselves, as well as in the Great Lakes, and trying to reduce any potential ecological and economic impacts that could occur from these invasive species get in there, like hydrilla. We also are, the main goal that we're also trying to do is to significantly reduce the risk of the hydrilla spreading from where it's already been found, and then potentially eradicating it in those areas where it's possible to do that. This is just a slide that talks about those different methods that oftentimes can be used. As I said, with hydrilla, it gets a little bit more difficult given the fact it highly fragments. It's easily transported by boats, as well as it's been documented wildlife can do that at times. So, and the type of system that we're in, grass carp and that are not as selective, and also not, we're dealing with a more open system here in Cayuga Lake, especially because it's connected to the Erie Canal, which then connects to all the waterways, many of the waterways throughout the entire state. So with that results, we kind of really centered on known EPA registered herbicides that have been used in other areas successfully. But as I said, because we're dealing with a high mixing area and an area that's really difficult, we're trying to treat hydrilla, keep it at those low levels so it doesn't have those ecological or economic impacts, and hopefully eradicate it where we can with safe and well-known herbicides that have been used. The two herbicides that we have been using since 2017, when we initially started in the Wells College Bay area, and these are herbicides that had been used back down in Cayuga Inlet during the original infestation in 2011 and 2012, is Sonar H4C, or at least sonar. We use a H4C version, which is a pelletized version that actually is a slow release. Sonar H4C is an herbicide that actually takes a relatively long time to actually kill the plants, 45 to 60 days of exposure of at least a lethal concentration. So it's not something that we just throw it out there and it kills the plants immediately, and then within a couple of weeks, we can evaluate that. This is a lower dose herbicide that actually impacts the plant over time and greatly reduces that. Because of that, and because there are varying susceptible or tolerances of other plants to the herbicide, in that we can dial in on concentrations of that herbicide to try to knock out hydrilla and reduce impacts to some of the native plants that we have out there, to try to reduce and make it a little bit more selective when with what we're doing. That, in addition to the fact that when we are applying these, we are applying them to known areas where we have identified hydrilla in the previous year or in previous years, and we're actually utilizing those areas with a little buffer onto that, because obviously if you find one plant or you find a patch of plants, and that sometimes there's a little bit fragments and some patches outside of that, so we want to make sure that we contain that entire area. The only other, because of the fact that we are dealing in locations where we have potable water intakes, the Village of Aurora has a water intake. Mackenzie Childs has a water supply system as well up in their area, and then there are camps that still do utilize shore wells, and that we actually utilize herbicides that have been identified by both the US EPA as well as by the DC at the levels that we're treating at, that they're safe to apply in those particular areas. In the case of sonar, there really are no restrictions with regards to the application of that. It can be applied. It does not impact the fish or you can eat the fish that are in there. It has a much lower impact on those particular species, especially at a concentration of 20 parts per billion or less, which is what we apply at. We're only trying to maintain about a 3 to 5 part per billion level in order. That's been found to be lethal for hydrilla. So as a result, we're treating it much lower than excuse me, than what we usually see. The only plants that are potentially impacted by this are tomato plants and some nursery greenhouse plants that could potentially and that's if it's at levels higher than 1 part per billion. As I said, we're treating it like 20, so we are well below the drinking water standard, and we test not only the potable water intakes, but we actually test the water itself to make sure we're maintaining those levels and not exceeding anything with regards to that. And we're coordinating directly with the county health departments with that and posting our results, as well as with local DEC as well. The only other type of herbicide that we use here, which is deemed safe in and around potable water intakes as well, is a chelated copper version. This is a contact herbicide. It is a much more broad spectrum herbicide, and it has been documented, at least at lower levels and lower concentrations, to be the least impactful to aquatic species, especially fish, in this version. The other thing is the way that it's applied, and making sure that you're in smaller locations to give the fish an opportunity as the herbicide is being applied, to be able to avoid the area, swim away from it, and thereby it greatly reduces any potential impacts we have with regard to that. We use these in spot locations or in locations that are relatively small in size, usually an acre up to 5 acres max, but usually a relatively small location. It's a one-time treatment usually. It is used either in treatment areas where we have patches that are being very resistant and are difficult we see are resisting the fluoridone applications, and need a little bit extra help to actually either kill the plant or reduce the plant's ability to be able to put those tubers that Catherine talked about produce those tubers, which they usually do in the fall for the next season, or for put it into the tuber bank, which those tubers can, now they've documented, can last in the sediment for up to 10 to 15 years before no longer being viable, and they don't all germinate, or they don't all sprout the next year. So as a result, when you get a patch in there, it doesn't take many years before you've got a tuber bank that's going to take a long time to exhaust all those tubers if you're trying to extirpate or eliminate those from that area. We treat the harpoon. Max is we follow the label. Maximum concentrations are usually 1 part per million, and it can be up to 1 part per million. That's what we have done, and you only apply to the height of the plant. So if you have a 10 foot of water and the plants are 2 feet off the bottom, you would only apply the copper and figure out how much you needed in order to maintain 1 part per million in that lower 2 foot. It's like the version that we use is actually a granular form, so it sinks to the bottom and holds down there relatively well. I know a question that's come up in previous years and has been concern over potentially the fact that some of the herbicides, in this particular case sonar, has a fluorinated carbon, and there's been a lot of talk about PFAS in the media and in the scientific community, and dealing with drinking water and all kinds of stuff and exposures and what the potential impacts could be. In 2023, I wanted to highlight this because this is important for what we're doing. In 2023, the Biden EPA went through a public rulemaking process and clearly defined PFAS as not including single fluorinated compounds. This deliberate exclusion of single fluorinated carbons was based on extensive scientific evidence and public input demonstrating that molecules with only 1 fluorinated carbon lack the persistence and bioaccumulation properties that are commonly associated with forever chemicals. So what has been identified or defined as PFAS are these forever chemicals. The single fluorinated ones are not considered in that realm. EPA approved single fluorinated compounds, and they're not considered forever chemicals. Therefore, they are not PFAS, and they do not pose any risk of concern when used as labeled. As I said, we are using herbicides that have been around for a long time, 40, 50 years, have been approved by EPA as well, and the state has approved them as well. We follow the label, and in the case of fluoridone, we're actually applying at the levels either at the level or lower than the levels that the state requires in and around potable water intake. So that's that would be an application or maintaining a concentration of at least 20 parts per billion. And of our monitoring that we do in the water, and that at least in our areas this year, the maximum concentrations that we had throughout the entire season was 10 parts per billion in one particular area. So generally speaking, and especially up in Aurora, we didn't have anything over 5 parts per billion. So in Ithaca, we did have some areas that went a little bit higher, but we still maintained it at half or less than what the state allows for safe levels of the fluoridone. Okay, so I'm going to start with talking about Ithaca first. Ithaca is actually our good luck story, and actually has responded very well to the treatments that we've done. For those that have been on these calls over the past several years, we originally started actually doing applications off of Stewart Park. We also had applications out off of Tremaine State Park, Marine Park. And then we've had applications that have occurred because of patches that occur within the inlet itself. We also used to treat Fall Creek as well. Since 2018, when we started doing those treatments, we were treating over 50 acres to 80 acres of area. Over that time, as the hydrilla has declined, we have pulled back our treatments as we always try to do, making sure that we keep herbicide on these patches because of these tubers in order to try to exhaust that tuber bank. After we don't see any patches for several years, we pull the herbicide back and use it more sparingly, and in the areas that we still have issues with, or in high priority areas in order to reduce the chance of spread of hydrilla. So in this case, the boat launches a lot of times and the marinas a lot of times are ones that we kind of concentrate on, because if there are fragments that are moved around in that, those are the areas potentially where we might see hydrilla getting established and or being able a point where we might see it escaping and going to other areas depending. So last year we actually pulled back our applications to just 17 acres. This is the launch at Truman State Marine Park, and then we have the marina that is at Cascadilla, the outlet of Cascadilla Creek. And then along the shoreline, basically, this is the Cornell Crew boathouse. This is a embayment. There's a lot of boat traffic, obviously, with the rowing and with the coaches' boats and that. And then we also, right here at the Boatyard Grill, we've had some patches that have occurred here along this edge, right where the old auxiliary docks were, as well as across. So we reduced our applications down just to the areas where we've had patches recently, or we've had active patches still as of last year. This year we opted to go with a 10-week application, which is very similar to what we've done in the past. 2 applications at 20 parts per billion, which is the label for potable water. There are no potable water intakes in here, so we actually could have used a higher concentration, but we opted to go with just the 20 parts per billion for 2 weeks. And then for 8 weeks, we did 13.75 parts per billion, because remember, we're only trying to achieve that 3 to 5 parts per billion. And it takes a couple weeks for that to build up and then maintain that at least for that 45 to 60 days that we need to, in order to be lethal or to damage the plants to the point where they are not able to put on tubers for the next year. We did the applications weekly on Thursdays and started on June 26th and went till August 28th. This is the water sampling that we do. State always requires us to do a half mile from our northernmost point as well as our southernmost treatment point. So in this case, because we actually have a splitting, 6 Mile Creek and then the inlet, we actually have 2 southern points, and then we have a northern point which happened to be just about at the confluence of the inlet with the Cayuga Lake itself. And then we had locations that were at each of our treatment areas. Truman State Park boat launch, the Cass Park, this is Boatyard Grill. So these particular areas where we actually have had patches of hydrilla in the past. The water, we did weekly water samples, and Bolton Point also did water sampling as well. The Bolton Point water sampling results didn't have anything that exceeded 1 part per billion, and most were below 0.5. All the samples for the entire 10-week period, as well as we also did monitoring for the month after application, never exceeded 5 parts per billion within the treatment areas, with the exception of Cascadilla Creek, which ranged between 6 and 10. So that's this area right here, which has traditionally had a lot of patches, and with the initial infestation that they discovered in 2011, that actually was identified right along this area here. So it's one of the more historic areas that has had hydrilla for a long time. All the residues were below 1 part per billion, with the exception of the Cornell Crew Bay area, which was just over 1 by October. So we had residues actually hanging around this year because a little longer than normal. I think that's a part of that is because we had relatively dry conditions during this time, not really that stormy. It was warm as well, and we also had We didn't have a lot of flushing or anything happen during this time period. We did monitor all the way until the second week in November, and this dropped down to less than 0.5, which is, so we documented that we have no more residues that were basically above one part per billion as required by the state. The point intercept sampling, that's why I say this is a good luck story. Throughout the entire season, we only captured or found hydrilla in the southernmost portion of the inlet itself. There were no patches actually found anywhere else other than right here, which was a fragment. These green points are actually in August, and then we always go back in early August, and then we go back in the end of September to see at the end of the treatment, to see post-treatment really what's still hanging around and what are things that we might have to worry about. The only patch that we had actually remaining after September was this one patch here just off Boatyard Grill, which is probably a fragment that got established this summer from one of the patches that was along. This is where we were finding most of our hydrilla this year again, earlier in the season. So right now, looks like the treatments have worked really well. We've greatly reduced the amount of hydrilla here to the point where it's less than 0.25% of our point intercepts at any point in time. And then we also still have a good frequency of aquatic plants, including coontail, eelgrass, sago pondweed, whitestem. We also do get some invasive still. Eurasian water milfoil is still around and still one of the dominants. Not nearly as abundant as it was a while back, but we've seen a decline with that lake-wide, actually, not just within our areas that we've been treating. And then the area south of the Cascadola Creek are still going to maintain areas of concern. So you have the boat launch area, this area because of the marina and because we have boats in and out all season. And then you have this real high activity area, and we still do have some active patches as of this past year. So Ithaca looks great compared to what we started with in 2018, and especially what was started with way back in 2011. So that's a good luck story there. Liz, if you want, I can take a pause there if anybody has a quick question, and then I can move on to Aurora.
Sure. If folks would like to ask a question at this point, you can unmute yourself or raise your little Zoom hand, and I can help unmute you. All right. Okay.
Perfect.
Oh, there's someone
sorry,
unmute.
Sorry,
Karan,
this is Karan. I couldn't figure out how to raise my hand. Thanks, Rich. One question about the frequency of other plants in the inlet that you mentioned that looks relatively good in the point intercepts that you did. Is it possible to share the observations or the data on which that statement is based, that the frequency of the other plants is can we sort of put what we mean by relatively good in context somehow here?
Yeah, no, we'll include that in our summary reports as we do at the end, and then given the fact this year is the last year that we're actually actively participating in any or contracting to do any more applications of herbicides. We're actually shifting roles, given the fact this was a research type project. We're shifting our roles to actually doing more support of the local and the state, and helping them with the monitoring and development of their treatment plans. So we'll be doing a much larger analysis of all the data that we have, and doing a much larger summary. That'll either be published as a tech report, or it may be a peer-reviewed publication as well down the road. So we'll have the data in the summary reports. We'll also have we're taking it much further and going to have a lot more of that data actually analyzed more completely for a paper coming out here that's going to summarize our work that we've done in the two areas, basically.
Thanks. Would love to see that. Yeah, thank you.
Yep. Okay, so I'll jump into Aurora quick, and Aurora has been quite an interesting area to work on. When we first got out there in 2018 or 2016, 2017, they had just found it. Bob Johnson had done a lot of analysis at that particular point in time where we were finding patches that were growing to the surface, and I think the point intercept frequency was running around 50% finding hydrilla at those points. So there was a fair amount of hydrilla in a small 30-acre embayment here, and over time we've kind of concentrated on that area, but we've had this plant proving just how invasive it is and how difficult it is sometimes to eradicate, and the fact that it's hanging on in some less than ideal areas, and it spreads, and it's spreading kind of northward, has spread northward at least with fragmentation most likely, and the flows that have caused it to have small little patches that along this entire shoreline that's stretched from its original where we found it down in Wells College Bay, and now it's moved actually farther up past Ellis Point. So this is showing we've kind of split our original application area into a northern and a southern. The DEC takes care of the southern, and we have taken care of the northern. This past year, because we did have a little bit reduction in our funding, and herbicides and that don't get any cheaper every year, and neither does application of them. So we had to kind of pull back a little bit and really concentrate on what we felt was the most critical areas, which, as we talked with the fishermen and we've talked with other stakeholders, we've been trying to reduce the northward movement of this to keep it away from that northern end of the lake, which is highly valued. It's also, if hydrilla gets in there, it's going to be much more difficult to treat because of the amount of area that is potentially suitable habitat for hydrilla up in the northern end. So we've concentrated. This is Ellis Point right here. We found patches in the past just around that, so we've done that. There was originally a fragment found up in here. We did an application for a year, pulled back, and tried to stay down in this area. We've been having patches pretty consistently in this. I believe the fishermen call it Airplane Bay. We call it Mackenzie Childs area cove. It's basically the area of Mackenzie Childs is right over here, and it's that embayment that is just south of Ellis Point. This past year, or in years past, we actually did have a fluoridone application that kind of went along this area just due to trying to reduce those and pull back a little bit. These patches that we had last year were kind of well defined, so we actually decided to do a copper spot treatment in those areas this year, while still monitoring it to see how effective that was as compared to the larger fluoridone applications that were. Originally, we had patches that were strung in between here, so that's why we treated the whole length. We pulled back and reduced that. So last year, I think we did almost 100 acres of fluoridone. This year, we only did 72, and the copper treatment is a one-shot treatment. There were 4 areas that ranged anywhere between 0.8 and 1.7 acres for a total of 4.7 acres. This is a one-time application that was done on August 6th. The fluoridone applications. You remember the last one I said it was 220 part per billion, and then 13.75. What we've been finding is that if we can try to front load in these more higher mixing areas with higher concentrations, allow it to build up, that in those earlier times as the plants are just emerging, that's a critical point in there, and hoping that we can damage the plants and reduce the chances of them being able to survive, increasing the lethality, and possibly just weakening the plant to the point where it can't put as many tubers out, or any, hopefully. So this one was 20 parts, a 15 part, and then 6 12.5 parts. Still the same amount of herbicide being placed out over 10 weeks. We just adjusted how we were applying it and front loaded it a little bit more. The water monitoring, we had water samples that were collected weekly, just like we do at Ithaca. These are the water samples that we had. We have water sample stations placed every half mile along these contiguous areas, and then we do the half mile north for the northern point, and then you'll see there's a southern point that we take care of that's way far south. This is a half mile south of the DEC's treatment area that occurs down in this area. So we had all finished drinking water for the water plant at Village of Aurora was less than 0.5 parts per billion, so we didn't have any detection of any fluoridone in those samples. There were 4 samples from the Kenza Child water supply that were in the 1 to 1.2 parts per billion area. As I said, this is 50 times less than what is considered safe by New York State, or they allow they allow up to 50 parts per billion. With this is 1 to 1.2, but this was enough in which we did work with the state. We also worked with the health department to notify them if they were doing any applications to greenhouse or anything like that, that the levels were reaching the point where they may need to worry, or not worry, but they need to be aware of it. And like I said, there was 3 points that were at 1, which is just the minimum amount. And then there was one weekly point that was at 1.2, and then all the rest were less than that, or less than 0.5. Concentrations within the treatment areas ranged anywhere between 1 to 5 parts per million, with the majority of them being less than 2. So this is an area that gets a lot more wave action, a lot more wind. It's not nearly as protected as Cayuga Inlet, and so this is to be expected. The fluoridone, or the Sonar H4C, is a pelletized version, stays on the bottom, and that's really where the plant takes it up. As it's actively growing, it's taking it up that sediment water interface. So that's why we use that pelletized version in that slower release to try to do that. So we have a lot of times found that even though we're measuring at a 1 part per billion, that we're still seeing damage to the plants. And we were seeing that in our surveys that we did in August. It looked like most of the plants that we saw were of the hydrilla that we saw were actually showing damage from the fluoridone, which basically kills the plant by reducing the plant's ability to be able to produce a carotenoid that protects the chlorophyll in the plant. So the plant over time basically loses its chlorophyll and bleaches out, and then it no longer can produce food, and so it dies. We did monitor this one, and due to the higher mixing that we had, all sites were at less than 0.5 part per billion 2 weeks after the applications ceased. So actually it's 3 weeks, because it was 1 week is the last one that we do, and then this was 2 weeks after that. So a total of 3 weeks after our last application, we had no detectable fluoridone within the entire treatment areas. The cumulative point intercept sampling, we were doing great into August. We actually we had some plants that we were finding plants within the treatment areas, not a lot outside except for down here, which we expected we might see. You can see where this is where we had our spot treatment, and there was a couple that were found in the August samples. And then we had a bunch that were found in here within our treatment areas. But by the time it got to September, we didn't see as many. We did find a few here. So if you look actually where we applied the herbicide, we did a pretty good job of actually knocking back. The problem is that the plant is, because we're knocking it out of its ideal areas, it's showing that it can find other areas that it will hang on, and they're in the deeper areas of the plants. A lot of times we've noticed with divers helping us with this, there's a harder compacted area that the native plants don't seem to like. They kind of, there's like a narrow area where the the shallower plants drop out, and the plants that can handle deeper water like coontail and now starry stonewort, it's coming in. There's an area where it's kind of open and it's not ideal, but the hydrilla is actually surviving in that area at times, and it's growing kind of along the bottom before it then sprouts up later in the season. And what we found was in August, we saw really good impacts on this. We didn't see a lot of detections outside. That's the green here. And then when we came back in the fall, in late September, we actually found that there were some patches in Wells College Bay that had gotten going again. So they're going to have to be applications there, because these are pretty good sized patches they were pretty good sized by the time we got to September, because of the fact this plant can grow very rapidly in warm growing conditions. It's one of the fastest growing plants in biomass wise. And then we found some fragments that must have gotten established north of Ellis Point. And these are points right along, this cove, this is Great Gully, and there actually is a point in there as well. So we do have some small patches that need to be addressed before they start becoming much larger patches like we saw down here earlier in the treatment areas. Again, same thing that we saw down in Ithaca. We had good frequency of occurrence of SAV, including eelgrass, coontail, white stem pondweed, sago pondweed, elodea. Starry stonewort and curly pondweed are dominant, but at different times of the year. Curly leaf tends to be very dominant early in the season and then dies back as other plants begin to come on. Starry stonewort is there, but it becomes much more dominant in the fall and becomes really the dominant plant in the deeper water and moves up. It's oftentimes only found with sago pondweed and with coontail. I know a lot of fishermen have found that sago pondweed, which resembles almost like a grass. It's really thin. It's a narrow leaf pondweed, and a lot of times will wrap around your prop in that, almost like fishing line. It used to extend into September before it would really die back. And due to the warmer summers that we've had in the past few years, it's starting to go through its normal cycle, seeding, flowering and seeding out, and then dying back in August. So when we go out in September, what we see is that sago is still there, but it's not nearly as dominant as it was earlier in the season during the middle of the part of the growing season and stuff like that. So you're seeing the seasonality in the aquatic plant community, which has always happened. I think it's just there are some changes that are going on that are happening, not just in the areas where we're obviously doing our applications, but obviously this is a more regional type thing that's happening. And I know some of the fishermen have mentioned similar things are happening over on Seneca Lake, where there's no applications going on for hydrilla or that, but they have similar changes going on there. So there are definitely some things going on that we can't answer with the data that we're doing, but we do collect the data both in and out. And that's one of the things I would say is our no plant point intercepts, where we find no plants. Those are pretty stable throughout the year. So and those are including both the treatment areas and the non-treatment areas. So we know that we're impacting plants within the treatment areas, but we still see elodea, we still see white stem, we still see sago pondweed, elodea handling within the treatment areas as well as outside of the treatment areas. We still see a range of plants. So, and then this is something I'll hand off to to Emily. This is actually the overall hydrilla locations for both for all of the Aurora areas that we do, which extends well past Long Point and then well past Ellis. And these are all the locations where we found hydrilla this year. And just like we were finding, it looks like up in our area, DEC had pretty good control within the areas they're treating, but we're starting to find some patches now that are kind of skirting on the outside of where we've been applying in that, because these plants are obviously finding ways to survive in these less than ideal places where they originally were. Our path forward, like I said, is complete our data processing and analysis. We're going to prepare our post-treatment summary reports. And we should have those done here this spring, and we'll be combining those also with the 2024 reports, and everything will be out. And then I believe the Cayuga Lake Watershed Network has all the previous ones, while we're still trying to get the Hydrilla Clearinghouse website taken care of, so we can get that information back up and live. And then the last thing is as I said, we are due to funding and due to the authority we're under, we have reduced funding now, so we are no longer actively doing applications or treatments. Those have been passed off to the locals, and we're staying in for several years to help them out with tech support, as well as with any of the monitoring. Not just in this year, where we should have some funding to be able to do it next year as well. And that's it.
Great. Thank you so much, Rich. It's always funny because when you refer to local, sometimes for y'all at the federal level, that means the state partners too, right? And so we're all just like looking up and down
state
the
and local.
spectrum.
So absolutely. Absolutely.
Yeah. Okay, great. So Emily can go ahead and get her slides loaded up here. And if anyone has a follow-up question for Rich, we can take it now. All right, hearing none. Oh, okay, we've got Dave Fitzgerald here. I'll give you an unmute, Dave.
Yeah, just one quick question. Looking at your graphs and maps and whatnot, I am new to a lot of this, so you're aware. But all the focus seems to be on that area that is south of Mackenzie Childs, south of Aurora. Has there been any detection of hydrilla in the more northern parts of Cayuga Lake?
So that's the great part about this partnership that we have is that DEC, Finger Lakes PRISM, the Corps, we all do sampling, not just in our areas, but I know Finger Lakes PRISM and DEC especially, they do a lot more surveys all around the lake proper. And we also look to those citizen scientists and those landowners that are very knowledgeable about the lake, and they see something that doesn't look right, they give us a call and we check it out. So we have had some hits, at least because of the boat stewards. I think there was one found after a fishing tournament up on the northwestern end of the lake. But we went back out, did some searching, could not find anything. And so far, right now, knock on wood, we have not found anything farther north than what we found on the eastern shoreline. So we've been looking both east and western shoreline, and the eastern shoreline has been the one that predominantly has more. We found more hydrilla on than the western end. Although we do have a patch across that Emily's going to talk about that they treat on the opposite side of the lake.
Thank you.
Yep.
Okay, wonderful. Thank you, Dave. So now we'll move on to Emily Timke-Bensinger's presentation for the Department of Environmental Conservation.
All right. Everyone can hear me, right? Perfect. So yeah, I am Emily Timke-Bensinger. I am the Aquatic Invasive Species Coordinator with New York State DEC Region 7. So that kind of includes the Eastern Finger Lakes. The dividing line between Region 7 and 8 actually runs right through the middle of Cayuga Lake. So lucky me, I actually get to do 2 projects in 2 regions. I also work with the New York State Water Resources Institute out of Cornell University as well. And I am Just going to basically give a quick overview of our 2025 field season summary, an overview of the history of hydrilla in New York State, and discuss a little bit more about our hydrilla monitoring methods as well as control methods, and then get into what DEC did this past year as far as hydrilla control. So really quick, I just want to give a huge shout out to my summer crew. I pretty much everything that is listed, this awesome list of accomplishments could not have been done without them. So in Cayuga Lake alone, we monitored over 630 acres of Cayuga Lake, which is about 1 square mile. So we're close to one, I think I looked it up, 168th or 9th of the lake. So we're getting there. And then we also monitored another additional about 100 acres throughout the region. This year, I was able to do some applications of herbicide for hydrilla. So I am a certified applicator in category 5A, which is aquatic vegetation with New York State. So myself and my crew, who were then apprentices underneath me, were able to do these applications. Additionally to doing work on Cayuga Lake, we also monitored over 40 miles of river throughout the region and removed over 8,000 pounds of water chestnut. Okay. So just a quick overview of hydrilla in New York State. So it was first discovered in New York in 2008 in Creamery Pond and downstate, so Long Island, downstate. And then in 2011, we had our first detection of hydrilla in Cayuga Lake on the inlet in Tompkins County. And then we've had a handful of other detections throughout the lake that Catherine kind of outlined earlier. And our most recent detection that we've had in the state or newly detected area has been in the upper Susquehanna River, Tioga County. So that is near the town of Owego, just to be a little more specific. And so this is just a map of the general distribution of hydrilla within New York State. So I pulled this map from IMAP Invasives, which is the New York State Invasive Species Database. So if anyone is interested in looking at the distribution of hydrilla in the state, as well as any other invasive species in the state, I definitely recommend checking out IMAP Invasives. It's also an awesome way to report an invasive species if you think you found one, if you are unsure of exactly who to contact regarding a species, or if you're not sure, you can upload a photo and someone like myself or another expert will confirm it. And it's a free public database as well. Okay. So to get a little bit more into our monitoring efforts or monitoring methods, so Rich kind of covered most or how we do our monitoring, which is using a point intercept survey. And so I guess I also just want to touch a little bit on how we determine what sites are a priority to monitor. So typically that will include a site that is either has already had a hydrilla population discovered, that we've either been actively treating or that we haven't seen any hydrilla for a couple of years, but we still want to keep an eye on it. And then we'll also look at sites that have that are usually close to boat launches or high traffic areas, because those have a potential to bring hydrilla in and out of the water body as well. So we use what's called a point intercept survey for submerged aquatic vegetation. And the way we do this is we take a grid and we'll lay it over our designated area. We'll usually do a 50-meter grid for our early detection site. So a site where we haven't seen hydrilla, but we're just curious, or we want to check it out and make sure we're not seeing anything. And then we'll do a 25-meter grid for our known hydrilla locations. So those points are going to be much, much closer. Basically, we're doing that so we can really make sure that we're finally delineating our hydrilla patches so that when we do go to write up our control plans, we're doing that as best we can and covering what we need to. And we record so we do at least 2 rake tosses, or DEC does at least 2 rake tosses at each point. Army Corps will do 1. There's pros and cons to each. So we do at least 2 at each point on either side of the vessel. So, usually the right and left side of the boat to make sure we're covering as much area as we can. And then we will record our total vegetation density and our individual species density as well. And those are recorded on the following scale. So 0 meaning no plants, and then trace, sparse, moderate, and dense. And I have a photo. Actually, you can see this photo down at the bottom. This is one of my interns this past summer. She has what we would call a very, very dense rake. And I believe that was all Richardson's pondweed, actually inside of one of our treatment areas in the Lansing area. So if you take a look at that map, you see all those points. That's kind of our grid. We did a combination of a 50-meter and 25-meter grid. We did our 25-meter grid where we knew we had hydrilla, and then our 50-meter grid in the surrounding areas, just to, again, double check those. And then the darker the points, that usually means the denser or the more vegetation that we're finding. This is just a photo visualization of what we mean when we're talking about trace, sparse, moderate, and dense. And then, like Rich said, we also record our biobase or our acoustics. So we have a transducer on the back of our boat. It's pretty much a fish finder. And we use that to record all that following information. And then we plug it into this fancy computer. We upload it to online, and it's processed for us. And it spits out these really nice colored, very pretty, what do you call it? Rasters that show the different vegetation densities throughout our survey area. So the reason we do this is because obviously when we're doing a point intercept survey, we can't totally cover every single square foot of the lake. So this just gives us an insight as to, what else we might be seeing, or what other, how dense our vegetation is. So these maps just review our monitoring locations for 2025. So as I said earlier, this year I was doing some of the applications, so we weren't able to monitor quite as many sites as we had in previous years, but we still were able to get out and cover all of our treatment sites, as well as we went up to Cayuga Island in the north end of the lake, and we did a survey up there as well, just to, again, check it out, make sure we weren't seeing any hydrilla where we don't want it to be. And then I have another map of all of our hydrilla detections that we had in the lake. So this does include the Army Corps' information, DEC's survey results, as well as the Finger Lakes PRISM results that have been shared with me. And then in the middle, this is our graph of our relative frequency of relative abundance of species at vegetated points. And this is specific to the Sheldrake area. So that's that spot all the way to the west on the lake. So basically we'll record what species we're seeing, how densely we're observing those species. And then we will graph it and just use that to kind of visualize what we're seeing. And those percentages, those basically mean anytime you throw a rake, for example, we've got a 36% chance that we're going to be pulling up Eurasian water milfoil. So just to give everyone an idea of what that means. Okay, so we also have our invasive species control strategies that are used not only for hydrilla, but to also manage other invasive species within the state, as well as on multiple different levels. So some of those might include no management, physical control, biological control, or chemical control. And I'll talk a little bit more about each of these in a minute. So whenever we're using a combination of 2 or more, we would call that integrated pest management. And that's kind of what we are, trying to, or most invasive species projects will use some form of integrated pest management. So first off, no management. It's a pretty easy one to do. You don't do anything. Existing populations will continue to grow and spread to new locations in Cayuga Lake and other water bodies, and surrounding water bodies would be at risk of spread for hydrilla. So Catherine showed us that map earlier showing all of the locations that folks are taking their boats out of Cayuga Lake and to other spots and water bodies within New York State. And then to also mention that at the northern end of Cayuga Lake, it's connected to the Erie Canal via the Seneca-Cayuga Canal as well. So if hydrilla were to get into that canal, it would be bad news. So with no management, we would probably have to increase our education and outreach in Cayuga Lake and the surrounding area, just to raise awareness about hydrilla and other aquatic invasive species to help prevent the spread of these species by human activity. Unfortunately, though, each season hydrilla will grow into dense mats that will outshade and outcompete native plants. And this option for management, it is used in some smaller private ponds with little or no connection to other water bodies. And the reason we do that is because we just, we have limited resources and we don't always have the funding to control hydrilla in these small private ponds that have little risk of spread. So again, low initial costs, but it can lead to future economic loss and negative recreation impacts. So again, thinking about those photos that Catherine had shown earlier of those super dense mats of hydrilla. Physical control. So there are a handful of different physical control methods, such as mechanical harvesting, benthic barriers, diver-assisted suction harvesting, or dash, or hand pulling, and dredging. So just to start out with mechanical harvesting, it's not recommended for hydrilla. So hydrilla spreads by fragmentation, and it can be somewhat brittle. So when the harvesters come through, they kind of chop up the hydrilla and can potentially spread it to other locations within the lake. Mechanical harvesting, it can be used for other invasive species, but just not in this case. Benthic barriers, so they can be used successfully if the conditions are right. So a benthic barrier, it's a heavy mat or tarp that's placed on the bottom of the lake over the target area, and then it's secured. And it works by blocking sunlight to plants. However, it will only work in certain areas, maybe where there's a low current or low wave action or no wave action, ideally, or low flow. And it also has to be in a relatively shallow area because these benthic mats need to be installed by people. And, we have to be able to basically wear like chest waders or something like that to be putting them in, or hire divers, etc. And it can be very costly. And typically the benthic barriers will only work in areas that are less than 1 acre. So again, not always ideal for larger patches or some of the patches that we're dealing with in Cayuga Lake. And not only to mention that these tarps, they need to be removed at the end of every year. So DEC, that's a rule by DEC, or that they have to be installed in the spring and then they have to come out in the fall. And then these tarps, they're not selective, so anything that's underneath them will also be killed as well. Diver-assisted suction and harvesting, again, not recommended for hydrilla, but can be used for other invasive species. So basically, scuba divers use a large vacuum to suck up material, and it requires specialized equipment, which usually requires a contractor that has access to this equipment. And it's not always the most selective, and it's not super great for large areas. And the suction harvesting, it doesn't suck up the tubers that can be in the sediment. So, if you remove that plant biomass, but you're not doing anything about the tubers, the hydrilla will just continue to grow. So in the past, it has been used in the Cayuga Inlet, but it was found to be ineffective for control purposes due to the breakup and dispersal of the brittle plant matter. So again, it is something we have tried. It just did not work out. So dredging. So dredging actually works super well in small areas. So we've had some success at a location on Cayuga Lake in Kings Ferry. So again, it can work, but it typically needs to be in a relatively small area, such as a boat launch or marina, somewhere where it's easy to get equipment in and out of. And then dredging, it also on the downside, it can be relatively expensive for the area that you're able to treat. And you also have to find a location to dispose of spoils as well. And it's also not selective. So when you're dredging, you're going to basically just dig up everything that's there, including a native seed bank as well as the hydrilla. Biocontrol. So I know that that's something that is usually, or folks usually will ask me about, is when is there going to be a biocontrol? Unfortunately, we don't have any biocontrol agents that are currently available for hydrilla. So the process to have an approved biocontrol, it's long and it's rigorous. Just to ensure that any biocontrol that is used is not going to cause any further damage to our ecosystem. So that would be kind of an example if anyone is familiar with the Australia cane toad story. Definitely pretty interesting. Check it out. But, you know, we're trying to prevent that. We don't want that to happen. So grass carp have been used for hydrilla control, but they're also not super selective and they can eat native vegetation and outcompete native species as well. So we don't use grass carp. They're also, just because of the size of Cayuga Lake and its connectivity to other water bodies, grass carp is not an option. All right, so that brings us to chemical. So it can be effective at low doses and is relatively selective depending on the herbicide that you're using. And it is ideal for our large scale infestation. So when we're trying to cover a large area with a fairly limited budget or resources, chemical is usually something that we'll turn to. And so for chemical applications, we do also have to apply for additional permits, such as our Article 15, which is our aquatic pesticide permit, and an Article 24, which is the DEC wetlands permit, if that's applicable to the specific area that we're applying to. So when we apply for a permit, DEC, we have to permit ourselves. We have to follow all the same regulations that all other organizations or anyone who wants to apply aquatic pesticide in New York State have to follow. So these are permits. They go through all of our natural resources folks, fisheries, Bureau of Ecosystem Health. They all take a look at it and, you know, flag anything that they think could be concerning. So the pesticide can only be applied by a certified applicator in category 5A or someone working under them as an apprentice. So someone such as myself who is a certified applicator. Oops. And so another part of the Article 15 permit or the aquatic pesticides permit is that we also have to notify our riparian owners. So anyone who lives along an application area that we had, you probably got a nice letter from me sometime in this past spring, just letting you know that we are doing a pesticide application in or around your area. And if you have any questions about that, to contact us or to contact the pesticides office. Okay, so again, just to review some of what Rich had talked about the herbicides that we used. So this year, DEC used Sonar H4C with an active ingredient of fluoridone. It's a selective systemic herbicide. So it acts by inhibiting photosynthesis and causes the affected plants to turn white or have chlorosis and eventually die. And we will typically apply a low concentration over an extended period of time for it to be effective. So to the right, you can see our application rate schedule. So we started out doing 20 parts per billion the first 2 weeks to try and, really hit those plants hard. And then we reduced that the following 8 weeks. So the fluoridone, it's taken up by the roots of the vegetation, then travels to the other parts of the plant. The herbicide act, and it will eventually kill the plant over a long period of time. And it can eventually eliminate the below ground part of the plant, including those tubers and turions. And then this year we also used Harpoon granular, which is that chelated copper herbicide. So it's a contact herbicide, and we used that as a spot treatment to control hydrilla. So we determined what spots we were going to treat during our, usually during our summer or that August monitoring event. And again, like Rich said, we usually will use that as a follow-up treatment to control maybe some stubborn plants or plants that we found just outside of our treatment area or out of our sonar treatment area. So I'm going to also just talk a little bit about some of the hydrilla control efforts that DEC had or put in this summer. So first, we have our South Aurora area. So that is just south of the North Aurora area. And this was applied by a contractor, which is part of a 3-year contract. So this contract will be ending this coming year. And we will, once that's over, we'll be adjusting some of our treatment plans. And so the contractor applied sonar once a week for 10 weeks to maintain our desired concentration, and then water samples were collected 24 to 48 hours after the application and taken to the Community Science Institute for analysis. And then we also had a Harpoon granular application as a spot treatment in about 7.5 acres at about 1 foot of plant height. In this area, it was monitored by DEC, Army Corps, Fish and Wildlife Service, and Finger Lakes PRISM in June, August, and September. And I just want to say that all of our water sample locations and results are posted on my program website, or the Region 7 Aquatic Invasive Species Program website. At the end of the presentation, I do have a QR code and a link available. So if anyone's interested in those, you can definitely check them out. So we began these applications at the end of June. So I did end up doing a couple initial applications, and this was just due to a shortage in herbicide supply and with some procurement issues that we were having. And then the contractors picked up those applications 2 weeks later and concluded on the 9th of September. So I treated a couple small select areas, and then the contractor came and also treated in those same areas, but it lagged a little bit. So, those areas I started out treating, they didn't treat towards the end, just to make sure we were staying within our permit guidelines. So here are just some maps of our treatment areas. So all the way on the left-hand side, those are our Harpoon granular application areas. So that was we determined those spots based off of the August surveys. So pretty much anywhere you see a little yellow circle or square, that's where we applied Harpoon, and that's where we found hydrilla. We also have in the middle, that is our fluoridone application areas. So that was that application that occurred once a week for 10 weeks. And then we also, all the way to the right, are our hydrilla detections from September 2025. So this is what we will be working on developing a treatment plan for in the very near future. To move on just to the south at Long Point, or just south of Long Point. So this was an area that I did the applications for, again, applied once a week for 10 weeks to maintain our desired concentration over that period of time. And we collected water samples 24 to 48 hours post-application and took them to the same spot to be analyzed. And then later in the season, towards the end of August, we did a harpoon granular application as a spot treatment for 2 small hydrilla detections that we had within our treatment areas. So if you take a look at the map, so there's that purple square, and then there's like a bluish green square. Those were our application areas. And then the 2 smaller yellow hatched circles, that's where we did our harpoon application. Myers Park Marina, Lansing Harbor, and Ladoga Bay. So I did the application in this area as well. We applied our sonar once a week for 10 weeks again, and collected water samples on the same schedule. And we also did a harpoon granular application in about 3.5 acres at an estimated 1 foot plant height out in the bay, and then about a 1/2 foot or 6-inch plant height estimated in the Myers Park Marina. So if you just take a quick look comparing our harpoon application areas and our hydrilla detections to our sonar application areas, you can definitely see that within our sonar application area in Ladoga Bay, we did have pretty good control. We didn't really see anything in there. Every hydrilla detection we had was kind of in the peripheral of our sonar application area. So I consider it a success in our application area. And then again, we're working to develop our plan for the upcoming year to make sure we can try and establish control over those plants in Ladoga Bay. So Sheldrake, again, this area we had an application by a contractor in this particular spot. Same application schedule. We were a little behind on our initial application, and that was mostly due to we were unsure if we were going to have funding to cover this spot, and we were able to, so things were just running a little behind because we still did have to go through that same permitting process. And sometimes that can take a little bit of time. So we also did our water sample collection after our application, and I don't know if anyone from Seneca County Soil and Water is on right now, but I want to give them a huge shout out. They were able to help us out with taking some samples due to a handful of scheduling conflicts that we had. So they were awesome in stepping up to do that for us. And then so the middle or the map to your left, those are our sonar application sites, and then the map to the right that shows our hydrilla detections pretty much from 2023 all the way to 2025, just showing that a lot of our detections were pretty concentrated in they've been pretty concentrated in some small patches along that northern shore of Sheldrake Point, as well as near the boat launch and within that little cove. So last year we did have a couple detections right by the winery or by that winery dock. But we didn't see anything down there this year. So again, I'm excited about that. Hopefully we don't see anything again in the future. And that means, we've at least contained it to just north of Sheldrake Point. So just looking forward, Army Corps, like they've said, will be stepping down from control efforts, but still assisting with monitoring and other technical items. So New York State DEC will now be the lead agency on the Cayuga Lake Hydrilla Control Project. And the Cayuga Lake Hydrilla Management Plan, which was written about now 6 years ago, will be updated to reflect the changes in control efforts and resource availability. So that's something that myself and some other folks at DEC will be working on. And currently DEC is putting together our finalized treatment plans for 2026. Hopefully we'll be able to share those pretty soon. I think right now we're working on getting them out to bid. So we'll see what we're able to share. And I believe that was, that was everything I had. So there's my contact information. If anyone has any questions, please feel free to reach out. And then there's that QR code as well as a link to the Region 7 AIS program website. I have my email with my name on it, or if you don't feel like typing all those letters, which sometimes I don't either, you can just reach out to me at r7ais@dec.ny.gov.
Great, thank you so much, Emily. So we have about half an hour, just a little less than half an hour here for some community discussion and Q&A panel. It looks like Charlie might have a question first for Emily, and then we can definitely open that back up and invite the other speakers to participate in that Q&A as well, or that folks can put out a question generally. For this portion for the panel, we'll just make sure that we give a bunch of people the opportunity to ask questions. So if I have to keep the conversation moving on, just understand that that's probably why. Okay, thanks so much. And I'll turn it over to Charlie for his question.
Hey, Emily, I saw on one of the maps that you had a red box around Cayuga Island, and maybe I missed it, but I didn't hear any commentary about Cayuga Island. Can you speak to that?
Yeah, so that was just our map of our monitoring location. So we only monitored up at Cayuga Island. We didn't treat or anything up there. So yep, we were just checking it out, making sure we weren't seeing anything.
So nothing was identified off Cayuga Island. Okay.
Nope, nothing up there, nothing of
Great.
concern up there.
Okay, thank you.
Great. Thanks. And then I'll also add it. I know there was a lot of detail on the slides that the presenters shared. So I'm going to try to get those posted online as PDFs by tomorrow morning, if not this evening, for folks so that you'd be able to go back and check those out sooner rather than later. All right. It looks like Karan. Yes.
Okay, thanks all. I guess I had a question about the plans going forward. So if this year also certain places were not treated that Hydrilla did exist in, and given resource constraints, it sounds like is the goal still eradication going forward, or is it sort of a more sort of quantitative management where it becomes a problem?
Yeah, so at this point, in some particular areas, eradication might not be possible, just based on, how widespread the hydrilla is. I think, again, we're kind of discussing how we might need to shift our management approach or our management plan. So we do have that Cayuga Lake management document that will be set to kind of renew and revive this next upcoming year. So that's definitely something that we'll be discussing, is how we want to move forward. But again, trying, it's a big beast that we're trying to control. And we're trying our best, but again, we are becoming what seems like increasingly limited with resources. As Rich said, herbicides aren't getting cheaper. Contractors aren't getting cheaper. So
and also just some of the biology. I mean, the tubers last a long time, and especially where there's a lot of dilution in the lake, it sounds like something has been learned about in a large water body like this, even scientifically. But or at least as far as I understand. But one thing if eradication throughout the lake is no longer the goal, then some of the arguments against mechanical methods where hydrilla is growing out of control, the fact that you might get some fragmentation that would spread seem less of an issue intuitively to me, because it's okay if there's a little bit of hydrilla. We just have to make sure that it's not getting out of hand in the areas where it is running amok. So does that, if the project is shifting away from eradication throughout the lake, would everybody agree with that? I mean, does that sort of shift the considerations of mechanical versus chemical methods considerably?
I'll jump in on that one, and Emily, you can follow if you want. I would still say that mechanical is lower, much lower on the you know the methods that we would want to use. The problem with mechanical is, first off, especially during the growing season, it has to be redone over and over because the plants grow back. It's like mowing your lawn. The plants come right back pretty quickly. The problem with that is that in areas where hydrilla may be not a dominant plant and may be just in the area but not really dominant, you're now setting conditions up to actually increase its potential impact and potentially its spread into other areas. This has been shown countless times, not just with hydrilla, but also with the Eurasian water milfoil and some other invasives that grow very quickly and grow from fragments. That definitely can skew can actually skew the playing field to favor those invasive really fast growing plants as opposed to the native plants, which don't grow necessarily to the surface, stay lower, are utilized not only by our fish and wildlife, but also are nicer for boaters and swimmers, as you're well aware. So yeah, no, I would still say mechanical is a method, but not necessarily would not rise up at that level. It's still considered lower even in those areas where hydrilla management is part of it, but they definitely don't. That one, I think initially for a while is going to be lower on the spectrum. So.
Right. And just to add to that too, we're also, maybe if we can't eradicate it, we're still looking to control it and suppress it as much as we can, just to, again, keep it from spreading to other areas within, other water bodies within central New York and even to other states as well. So yeah, we're mostly looking to contain, control, suppress, but yeah.
I'm wondering whether you're monitoring as far north as Union Springs, where there are often fishing tournaments. Are you monitoring for early detection simply, or off Taganic? I don't know if you're monitoring there either. I remember at the beginning, Bob Johnson didn't know how far up the lakeshore he should go. And I think we convinced him to go maybe as far as Myers, but I'm not sure he did that religiously. But if you find it very early, you have a chance at least. So are you monitoring at the northern end where there are boat launches?
I can speak to this from the Finger Lakes PRISM perspective. So we do routine monitoring on a yearly basis. And this past season, we did monitor at Union Springs as well as Mudlock, which is the tip of Cayuga Lake. And so that area we're also particularly interested in, making sure that there's no hydrilla there because of the connection over to Seneca. So we do monitoring at the northern end, and I'm trying to work in continued monitoring up there as well for the following season this year.
Great. Thank you.
You're welcome.
Yeah, and I would also add that I know we had a potential hit with the one of the stewards at Cayuga Lake State Park launch, and after a tournament, and before the harvesters went out, the Soil and Water actually contacted us, all of us, and we went out and actually did a survey along the areas they were planning on doing harvesting to make sure that there was no hydrilla in those areas, and we didn't identify any. So that's the great part of this partnership is that there is a lot of communication that happens for either that early detection or to avoid potentially spreading inadvertently with some less than ideal techniques. So.
To me, it's surprising you don't find anything off of Taganic, except it gets very deep, very fast there.
Yeah. We've monitored at Taganic in the past. This year we didn't, just due to some schedule constraints with having to do applications in the Myers and Lansing area. But yeah, it gets super watershed or super deep, super huge drop off, but we do try and monitor down there on at least a biannual basis.
Thanks for that question, Elaine. Looks like Karan's got another one.
If I'm not stepping on anybody's toes.
Go for it.
I had one more, again, related to this issue of effects on other plant species and what we know about that through the lake. I was reading a little bit recently, and this may be known to some of you actually, but I mean, it turns out that fluoridone is many of you probably know this is used to treat Elodea in many in some lakes in Alaska, right? So Elodea is considered invasive in Alaska. And I was struck by how similar some of the description is for the treatments there as compared to here. So Elodea is, of course, one of the important native species that I think we've seen. There's report about the frequency from what Emily shared. But I'm looking at a slide from the Fish and Wildlife Service talking about this, and they say that fluoridone kills Elodea systematically and selectively with few non-target effects. Absorbed via roots and shoots, needs to be in the water column for 45 to 90 days at concentrations of 4 to 10 parts per billion. So, I mean, and applied, I mean, they're using slow release pellets there as well. These are some smaller lakes, including Lakes Stormy and Daniels, interestingly enough. But the point is, the manner of the application, the concentrations are not very far off and very much in line with some regions that you guys are describing. How do we make sense of the fact that, it's described as selective to Elodea in Alaska, but should not affect Elodea, for example, here, and does not appear to be, at least from the sort of frequency of observation data that you show, right? I mean, we're not seeing biomass data, we're not seeing plant abundance, as far as I understand it. But what should I make of the fact that it's described as selective to Elodea out there?
So the selection that you're talking about there, remember, we're talking 3 to 5. We're trying to maintain, and we're usually less than that for killing hydrilla. And we're trying to maintain that 3 to 5 parts, which we do. There was only one area that we actually had it higher, and that was Cornell Crew Bay area, or Cascadilla Creek area, excuse me. That has predominantly been milfoil and does not have some of the other natives in there. So that's the whole way that you can do selectivity with some of these herbicides, is to find a range in which the plant is susceptible to it, but other plants that are around there are not, right? We know that. So they have a much different complement of plants in Alaska than we have here. We're actually looking at hydrilla and trying to hit it lower. So that's why we're using lower concentrations and trying to maintain that 3 to 5, so that if we get any herbicide we put out there, if we do it at a high enough concentration, can kill most of the plants that are out there. But that's not our goal. Our goal is to try to target, in this case, hydrilla, which has a much larger impact on biomass and potential ecological impacts than Elodea would have. Here we have one native, and there are non-native species within the lake that also occur. We're just not selectively going after those species. We're trying to do hydrilla, and that's where I would say the difference range. Yes, it sounds pretty close, 4 to 10 versus 3 to 5, but that range is the range that we're trying to do. And sometimes we go a little higher and we have to pull it back, but oftentimes we're a little lower. We're still seeing damage to the hydrilla. We don't see that same damage to allodia and even in the treatment areas. So there is a difference in toleration of fluoridone at the levels that we're using. So that's the way I could say that we're doing, we're trying to be more selective, is just by doing that narrower range. So.
Okay, I get you. Your answer is that it's strictly on the concentration, which, I mean, I would to me it's remarkable that a difference of even the difference between 4 to 10 and 3 to 5. I mean, there's obviously overlap there, and considering that we apply at 20 ppb and that decays over time, I don't know exactly the dynamics, and of course there's variation there, but I would love to see. Again, here, just given how small that difference in concentration is, if that's really the only difference in the selectivity, I would really like to see the evidence. How selective do we know that it is, if we're going to claim that it is? It's remarkable to me that the same chemical is selective to a different organism at concentrations different by a couple of ppb, or at most a few 10% in percentage, in a different place, of course. Anyway, but I thank you for the answer. It's about the concentration there.
And also remember that the formulation we're using, it's a 20 part per billion going out, but it's a slow release pellet. So it's the chemical itself, the herbicide itself, is being released over several weeks. So that
Yes.
we're applying it at 20 parts, but it's actually several weeks that it's going to release all of that 20 parts. So that's the other game we're playing there too. We're not throwing it out and it instantly spikes and decays. It's actually building up over time. And then that's why we're doing those weekly treatments. So as some are coming down, the others are coming up, but we're trying to maintain that. So.
Thanks.
Could I make a comment?
Yes, John, you're welcome to.
Yeah, I just wanted to make a comment. Oftentimes, a lot of, people have said that these weeds are spread by boat trailers. That's probably the number one way that they're spread. And I made a comment last year, and I want to make it again this year. I think the New York DEC and the state parks and the PRISM group really need to do something about the floating weeds around the boat launches. I don't know what the solution is, whether it would be to have some tools there and just give people the chance to clean the boat lanes out. But I know that at some of the launches, like Dean's Cove is an example, I pull my boat trailer out and it is just covered. Now, I know the water stewards are there and sometimes they have downtime and maybe they could help out. But I really think you guys really need to focus on getting all the floating weeds out of the boat launches in the the immediate vicinity of the boat launches, because I agree that's probably where most of the transfer is going, to other bodies of water. I just don't see much of an effort if cleaning out the boat launches it would probably have to be on a dang near daily basis from beginning of June through September.
Thank you for your comment, John. I think that's an interesting point. I personally don't run the Watercraft Inspection Steward program, but I work closely with the program manager who does. And it's interesting you bring this up because she is looking into the impacts of skimming the surface of the water and how that impacts the plants that get attached to boat trailers. And so it seems like a very logical, yes, if there's more plants, they'll get attached more often to boat trailers. But it's an important first step for us to just prove that mathematically and have it in writing that this is happening. And so there were some stewards last season who were doing those, like a skimming routine almost on a daily basis, that they were present at the launches. And I can't speak to whether it's going to be done again this season, but I will bring up this point to her that might be something that people are interested in at public launches. So I will.
Yeah, because a lot of the stewards would take the time to come over and actually help you pick off the weeds off of your trailer. And I just think maybe it'd be a better idea to just have some sort of large rakes or something, or skimmers that you could just have right there at the launch, so people could do it. I mean, I would gladly take 10 minutes out of my time and clean the launch that I just launched out of. And I don't know. I don't know, just something to think about. That's all.
Yeah, no, it's a great point. And we have started exploring the idea. So I will say that.
All right, thanks.
Can I say something? Years ago, when the infestation first started at Ithaca, Bob Johnson was studying after the season the incidence of the weeds that were the plants that were still available around the area that was being treated. So there's a lot of data available someplace, I don't know where, with his observations of the native plants or the plants that were available after the fluoridone treatment. Somebody from Ithaca must remember this.
Thanks, Elaine. I've read a fair bit of his reports, and I thought they were extremely valuable. And up to I think about 2018 or 2019, they were reporting surveys, and I have those. I'm not sure if they're still publicly available at the moment, given some of the sites that have been down. But I think that's the kind of data that I think. If we're concerned about other plant species and biomass, how well they're doing, is extremely valuable. So if you look at those reports and compare, I'll just talk about Elodea again. I mean, there are areas where you would be seriously concerned about drops in biomass of Elodea. Now, there's year-to-year variation depending on where the surveys were done in the year that can contribute. But some of the difference looks pretty dramatic if you look at the numbers. And so I've looked at these reports. And as far as I can understand, after 2018 or 2019, when the scope of our applications in Cayuga Lake dramatically expanded, beyond the inlet and the southern end of the lake, but to Stewart Park and in these northern areas, and the amount going into the lake has gone up dramatically. I don't believe there's been any such monitoring since about 2018 or 2019, which was the last report I know of from Bob Johnson. But indeed, I think that data is valuable. It's striking to me that we have not been doing that monitoring since the scope of the applications, dramatically expanded.
I know that Emily does a lot of monitoring as well, so she might be able to speak to this point a bit, but I will add that for the past 2 years at least, the Finger Lakes PRISM has monitored a lot of the overlapping treatment sites, and we do take into consideration biomass data of all the native species that come up on the rakes. And so this information is maybe not publicly available at the moment, but it is something that we have and we would be willing to share if you're interested in seeing that. And I hope to move forward with making it more publicly accessible soon. Because we have it, we might as well show others. But yes, we've done monitoring down in Ithaca at Allen Trayman, as well as Sheldrake and Lansing, and we have all that plant data. So it is here, and it will be made available in the future.
That's good to know. Thanks. I would actually be happy to. I don't know how to.
I think
I'll write your name down and try to find your email. Yeah.
The slides will be shared, so you can contact me anytime.
Good. Yeah. I think it's great if that would be publicly available, and if possible to see it, I would be very interested to understand what we do know. Thanks.
Yeah, just to add, so we do like a pre and post application monitoring typically. And again, we'll do that with Army Corps in those large northern, southern Aurora areas that extend past our application areas. And we also do our pre and post application monitoring in Lansing, Ladoga, as well as in Sheldrake. So we do monitor, we do collect data about our plant composition in those particular areas. Not all of that I don't believe has been made available. Right now I'm working on putting together a report for this last year, which again should be up on that website that I had the link to, or that I shared the link to on my presentation. So that should be up there soon though.
Yeah, and I'll jump in and add one more thing before the last question there. We actually worked with Dr. Johnson or with Bob on a lot of the initial stuff we did in Ithaca. We also coordinated with him heavily when we started the work in Aurora, because he was the one that did the initial surveys. And a lot of the points that Bob actually set up originally are still included in all of our grids. So we have actually expanded in some of those areas where Bob didn't do much, but just did little location stuff. And then I believe it was just a couple of years ago, Emily actually, it wasn't Emily, it was actually Emily's predecessor actually redid all of Bob Johnson's kind of southern Cayuga Lake analysis, both in August when Bob would always do it, as well as did it again in the fall, which would have been the September, October surveys to do that mid-season, and then also the post. And like Emily said, we actually go out and do before the plants are coming up. We do during the treatments, and we do post. And with all of our treatment areas, we're usually doing monitoring at least a half a mile north and south of treatments or of the last known locations of where hydrilla has been. So there is a lot of data on plant communities and that around those areas too, that we'll be summarizing as well, and including in a lot of our stuff. We have it. It's going to be summarized and put together, and we summarize them in the reports, but a lot of that stuff will be summarized more and made more available.
Would love to see it. Yeah, I know that there are probably personnel reasons and other reasons why those reports weren't being, or this data, if it was continuing to be taken, wasn't being shared after 2018 or 2019. But I think, yeah, getting it out there, being able to see, what actually is being measured, beyond the qualitative descriptions that we can talk about on a call like this, hugely valuable. So I very much appreciate that. Would love to see it. Yeah.
Liz,
Right.
I have one question if there's still time.
Sure, Casey, you can have the last one.
Sure, thanks. So we've talked with you guys as a group of fishermen a lot, all the groups, about the change in habitat through the rest of the lake. You guys have talked a lot about the hydrilla treatment areas, but we've brought up other areas such as Union Springs and the north end, and the change in natural vegetation that was present in those areas. And Rich mentioned that due to warmer summers, pondweed is dying off earlier and going away. So it appears that there's, a climate change that's impacting some of that. But you would think that on the backside of that, a late season grass like coontail or something would maybe come in and fill in on the backside of that change. Has there been any discussions to secure any funding, finding out with what's going on with the rest of the lake and the lack of natural vegetation in the areas that have traditionally always held it, such as Union Springs, Cayuga, and the north end?
Yeah, that's a great question. I think there's so many questions we can be asking about the vegetation community in the lake and how that impacts fisheries. And I have a lot of my own. So that's not something that we, the Cayuga Lake Watershed Network has pursued in the last year just because of capacity reasons. But again, as a community-driven organization, the more that you guys come to us with these concerns, and the more we can kind of rally resources together, I think we could look into opportunities to do something like that. It would be a bigger research program. It'd be great to either have agencies involved or PIs from some of the university or colleges in the region to really buy into that as a sort of priority project that we would seek funding for. So thank you for bringing that back up. Yeah, hasn't happened from the network. If anyone else has pursued this, please do let us know, because we'd love to work with you.
I will say that at the moment, I know DEC isn't currently looking at any of those particular changes, but if that is something that folks are interested and concerned about, like we can definitely bring that to the attention of, we have folks that it's their job to monitor and look at the Finger Lakes and water quality and stuff like that. So, you know, I'm happy to pass that message along and let them know that this is something that's important, or that people are concerned about, as well as, yeah, maybe potentially connecting with any PIs or universities that might be interested in studying this.
Yeah, I think, Emily, just the background of that is a group of fishermen, we felt that there was a danger that the hydrilla treatments were affecting other areas of the lake. That's what we've had really good communication with Emily and Rich and the folks at DC that were involved in the program before. I think you just came on last year, if I remember right. But it seems like what we're all learning is that is something separate, but it is a very visually obvious thing that is, potentially detrimental to the, fishing in the lake and all that. It seems to be something going on. We've brought up farm runoff issues, water quality, for whatever other issues could be going on, but it does seem something else is going on in the lake. Liz, we brought it up last year, so I just figured I would touch on it and ask if there's been any conversations. I know your group wouldn't pursue it, but there's people in the room that have potential to pursue it.
That's great. Thank you for bringing that up, Casey. That was a great point to end this conversation on just for today, and hopefully continue it more in the next few months and keep the momentum going. So I want to take a moment to thank our panelists again. Thank you to Rich, Catherine, and Emily for taking their time to share these reports with us for the year and to speak a little bit more towards how the agencies are approaching these issues. As promised, I will get those PDFs with all the nitty-gritty details posted up online so folks can look at them, zoom in, do what you need to do. I saw some squinting here on the call, so we're gonna get those available. And then thank you again for joining, right? It's community members like you that support this organization, that make it possible for us to bring together a lot of different perspectives on how to best steward our lake, and make sure that we're in this ongoing conversation with the agencies as well. So thank you for joining, thank you for caring, and we will continue this work. Have a great evening, everyone.
Thank you so much.
Thanks.